Everyone. Today we're talking about something that sits at the absolute foundation of pathology. What happens when a cell gets stressed and how we decide whether it's going to survive or die. Everything else in pathology, inflammation, repair, cancer, starts right here. So think of this as learning the first domino in a very long chain. Here's the key concept I want you to hold on to for this entire talk. A cell's fate is not random. It's actually pretty logical. Three variables drive the outcome: what kind of stress hit it, how long that stress lasted, and how tough that particular cell type is. A liver hepatocyte and a cardiac myocyte can face the exact same stressor and have very different outcomes. That's not magic, that's biology. Run through what actually injures cells. And I want you to think in categories, not lists to memorize. Physical things crush, burn, or freeze cells. Chemicals and toxins interfere with metabolic machinery. Infections hijack or destroy cells directly. Ischemia is huge in pathology. Cut off blood flow and you cut off oxygen, and oxygen-starved cells die fast. Immune-mediated injury is the body's defense system going rogue, and nutritional deficiencies quietly starve cells of what they need to function. When you see a lesion clinically, ask yourself, which category does this fit? This is the most important concept in cell injury, the point of no return. Up until a certain point, if you remove the stressor, the cell can bounce back. We call that reversible injury. But cross a certain threshold and the cell is done. It's committed to death no matter what you do. The two big events that signal that crossover are membrane failure, when the cell membrane can no longer keep things in or out, and mitochondrial collapse. When the mitochondria fail permanently, the cell has lost its power plant. Game over. Here's where it gets really interesting. There are two main ways a cell can die, and they look and behave completely differently. Necrosis is the messy death. The cell swells, the membrane ruptures, contents spill out, and the immune system has to come up and clean up the mess. That cleanup process is what causes inflammation. Apoptosis, on the other hand, is a controlled, programmed death. The cell actually participates in its own demolition. It shrinks, packages its contents into neat little bodies, and neighboring cells quietly eat them up. No inflammation. Think of necrosis as a building fire versus apoptosis as a planned demolition. Not all necrosis looks the same, and that's actually clinically useful. The pattern of necrosis gives you a clue about what caused it. Coagulative necrosis is the most common type. It happens when blood supply is cut off. The tissue dies but keeps its shape, like a cast of the original tissue. We call it the tombstone effect. Liquefactive necrosis happens in the brain, where enzyme-rich tissue basically dissolves itself, or in abscesses, where bacterial enzymes liquefy tissue. Caseous necrosis, that soft cheese-like material, is a hallmark of mycobacterial disease, so think Johne's disease or tuberculosis. Fat necrosis happens in and around the pancreas when digestive enzymes escape, and gangrenous necrosis in extremities can be dry, coagulative, or wet when bacteria pile on. Pattern recognition here is a skill, not a memory list. About apoptosis that students often miss. It's not just about disease. It's absolutely normal and essential for life. Your fetal limb buds start as paddles, and apoptosis carves out the fingers. The immune system uses apoptosis to delete T cells that would attack the body's own tissues. Every day, billions of cells in your body and your patient's bodies are quietly deleted and replaced through apoptosis. It only becomes pathological when it's occurring in the wrong place or at the wrong rate. Too much apoptosis, degenerative diseases. Too little, cancer. Before we wrap up, let me give you the framework I want you to use every time you encounter a lesion, whether it's on a slide, in a gross specimen, or on an exam. Ask these 5 questions in sequence. This is a systems approach. You're not trying to remember every disease that causes necrosis. You're building a logical chain. Stressor leads to cell response, leads to type of death, leads to pattern, leads to diagnosis. If you can move through this chain fluidly, you'll be far ahead of anyone trying to memorize lists. Here are your 6 big ideas from today. Don't try to memorize every detail. Instead, make sure you can reason through each of these if someone asks you a case question. The framework is what matters. Next, we'll move into cellular adaptations. What happens when cells are stressed but survive and change their behavior? That's a whole other fascinating story. Pause the video, read case question 1, try your hand at it, and then we'll work through it together. This is coagulative necrosis. The preserved architecture, those ghost cell outlines, is the hallmark. This pattern most commonly results from ischemia, where blood flow was cut off. In the spleen, think infarct. The proteins in the cells denature and solidify, preserving the scaffold even as the cells die. Great follow-up question to ask yourself, what could have caused ischemia in that spleen? Thrombosis? Torsion? That's how you chain lesion patterns to diagnoses. Question 2, try your hand at it, and then we'll work through it together. Caseous necrosis, soft cheese-like granular material. This is the classic pattern for mycobacterial infection, including Mycobacterium avium paratuberculosis, which causes Johne's disease. The cheese-like appearance results from a combination of cell death and immune granuloma formation. It's actually a mix of dead macrophages and debris. If you see caseous necrosis, mycobacteria should jump immediately to the top of your differential. Pause the video, read question 3, and try your hand at it. We'll then work through it together. The key distinguishing feature is inflammation, or rather, its absence. Apoptosis does not trigger inflammation because the cell membrane stays intact throughout the process, and the apoptotic bodies are quickly engulfed by neighboring cells or macrophages before contents spill. Necrosis always triggers inflammation because the cell ruptures and releases its contents, which act as danger signals. Clinically, this matters because if you see inflammatory infiltrate around dying cells, you're looking at necrosis, and that tells you there's an ongoing pathological process driving tissue damage. Pause the video, read question 4, and try your hand at it. We'll then work through it together. This is a great case for the reversibility concept. At the burn margin, cells closest to the heat experience irreversible injury. Membrane failure and mitochondrial collapse have already occurred, and those cells will undergo coagulative necrosis. Cells slightly further away may be in reversible injury territory. Their membranes are stressed, organelles are swollen, but if perfusion is maintained and further heat damage prevented, they can recover. This is why burn wound management focuses so heavily on cooling, fluid support, and preventing secondary ischemia. You're trying to rescue the cells sitting right on that reversibility threshold. Video, read question 5, and try your hand at it. We'll then work through it together. This is fat necrosis, and it's a classic finding in pancreatitis in any species. When the pancreas is inflamed, digestive lipases leak out of the acinar cells and into the surrounding tissue. Those lipases break down triglycerides in nearby fat cells into fatty acids and glycerol. The fatty acids then bind calcium ions in the tissue, a process called saponification, forming calcium soaps. Those calcium fatty acid complexes are the white chalky deposits you're seeing. So the lesion is actually telling you two things at once. One, there was a lipase spill, and two, there was enough local calcium to drive saponification. It's chemistry you can see with the naked eye.